Hello, biology. The purpose of this video is to give those students who are unable to keep up with the pace in class an opportunity to get them all finished up, or if a student was absent, to give them an opportunity to get the notes done in the first place. All right, 11-2, we start talking about probability, how to use a Punnett square, reminds you of the differences between dominance and recessiveness, heterozygous and homozygous. Without further ado, here we go. Also, if you are turning the volume down, I will be pointing at the camera to indicate when we are moving ahead to the next slide, so you can pause it if you are still writing or typing. Let's see, to do a Google Doc, you're going to need 10 rows, 2 columns. And if you are doing this by hand, of course, just draw a line down the center of your page. Please make sure to start your numbering at 17. And of course, we're going to be finishing the numbering at 26. All right, here we go. So probability is the likelihood that an event will occur or it won't occur. So for example, if there is a 75% chance that Paulus is going to be wearing Rio Raider gear, that means that there would be a 25% chance that he is not going to be wearing Raider gear, and that's probably about accurate. So whatever the probability is that the event will occur, you subtract that from 100, and that will tell you what the probability that it is not going to happen. Okay, a Punnett square, it's a tool that is used to predict allele combinations. Remember, alleles are simply the letters that we use when doing genetics problems. They just simply represent whether or not they have the dominant form of the trait or the recessive form of the trait. And of course, filling in the Punnett square predicts what possibilities we will have in offspring. All right, moving ahead. Okay, symbols. We use capital letters for the dominant allele. Remember, DOM stands for dominant. We use lowercase for the recessive allele. So as you can see in this diagram, and I really would recommend using Snippet or doing screenshots and importing these images into your notes. But we've got a tall. Of course, we have a dominant allele. You only have to have one dominant allele to have the dominant trait. This one's a hybrid. It's a heterozygous. Okay, so we've got tall still because, again, you only need one of the dominant alleles. And you only have a 1 in 4 or 25% chance of getting the recessive phenotype, which is the appearance, because you are getting one of the little t's from one parent and the other little t's from the other, and it's only a 1 in 4 chance of that happening. And of course, the only way that that can happen is if both parents actually have one of the little t's. If this parent was big T, there would be no chance of getting 2 little t's together and getting this shorter plant. Okay, the F1, remember the F1 stands for the first filial generation, the first generation of offspring. F1 parent gametes are shown along the top. And along the side. So these are the gametes from the F1 parents. These are the F1 offspring themselves. Oh, beg your pardon. Beg your pardon. We're talking about the self-pollination of the F1 generation, which were both hybrids. So the parents of these, one of them was big T, and the other parent was little t. Therefore, each of these hybrids are heterozygous. So these are the F1 offspring, and then he self-pollinated the F1 offspring to get the F2 generation. So these are the possible outcomes in the F2. So if you remember back to the notes from 11-1, When he had a tall parent and a short parent, all of the offspring were tall. That's because they had one big T and one little t. Then when he self-pollinated that F1 generation, the hybrids, you got a 1 in 4 chance of getting a short plant. Okay. So this is just taking that experiment and applying it to how you would use it in a Punnett square. I think if you remember correctly, back to the 11-1 notes, I said you could do that, where it was showing where the different letters went towards the end of the notes. You could do that in a Punnett square as well. Obviously, you can. Moving on. Okay, please use this as an opportunity to practice a little bit with your notes. You might want to take a look back at number 17 for this. The blank blank that might result from a genetic cross can be determined by using a Punnett square. Oh, beg your pardon. This would be number 18. You'd want to take a look back. Yeah, that would be the allele combinations or genetic variations that might result from a genetic cross. Genetic variations are going to be referring to the phenotype. The allele combinations would be referring to the genotype. Okay, capital letters are used to represent blank traits, while lowercase letters are used to represent blank traits. Is that recessive in the first blank, dominant in the second? Dominant in the first blank, recessive in the second. Excuse me. Yeah, that's dominant traits, capital letters, recessive traits, lowercase letters. Okay, onward. Some more vocabulary. Homozygous, we're going to abbreviate that from now on as HMZ. Homo is Latin for same. So organisms with the two same alleles for a particular trait, that can be homozygous dominant, big T, homozygous recessive, little t. Organisms are true breeding for a trait if they are homozygous, meaning they are not hybrids. Heterozygous, we're going to abbreviate that as HTZ. Those are organisms with two different alleles for the same trait, in this case, big T, little t. And those organisms, beg your pardon, are called hybrids. And you get hybrids when you cross a homozygous dominant tall plant, big T, with a homozygous recessive plant, little t. And all of your offspring will be big T, little t. And that is exactly what happened in the F1 generation when he crossed two true breeding parents. All right, moving ahead. All right, some more vocabulary. Phenotype, we're going to abbreviate as PT, the physical characteristics of an organism. For example, tall, short. Genotype, we're going to abbreviate as GT, the genetic makeup of the symbols, or the genetic makeup or the symbols is what I meant to say. That can be big T, little t, or little t. Those are all genotypes. Big T, and big T, little t are going to equal a tall phenotype, and a little t is going to equal a short phenotype. So as you can see, more than one different type of genotype can express the same trait. Big T, and big T are both going to express the same phenotype. Okay? Moving ahead. All right. Okay, so phenotype versus genotype example. Big T is homozygous for tall or homozygous dominant. Big T, little t is heterozygous for tall. You don't have to say heterozygous dominant. Little t is homozygous for short or homozygous recessive. Excuse me. As you can see, Big T, big T, big T, little t, which are different genotypes, both have the exact same phenotype. And the reason why that is as long as you have one big T, you will get the dominant phenotype. All right, moving ahead. All righty. So how are probability and segregation related? So remember back to section 11.1. Segregation referred to the fact that two alleles, remember all parent cells have two alleles or two chromosomes. When gametes, sperm and egg cells are made, it separates those two chromosomes, separates those two alleles, okay? The only way for the short individual in the F2 generation to come back is if each parent has one little t. And then, of course, during fertilization, both those little t's come back together, and you get a 1 in 4 chance. Okay? 1/4 or a 1/4 probability of the F2 plants will have two alleles for tallness or homozygous dominant. 2/4 or 1/2 have one allele for tall and one allele for short. Sorry, I should have grabbed a cup of coffee before I recorded this video. So this first example is going to be this guy. Second example is going to be these two. All right, so a 1/2 chance that they're going to be heterozygous, and then a 1/4 probability or a 1 in 4 probability that they're going to get both of the lowercase alleles. And of course, that means that they would have the phenotype of being short. Oh, I'm going to try to power through, you guys. I'm really sorry. I did not sleep well last night. All right. Moving ahead. Okay, ratios of offspring. So percentages and fractions are pretty easy to calculate and determine. Ratios are a little bit more complex. Lots of kids have been exposed to fractions and percentages, but not a lot of kids have been exposed to a ratio. So because the allele for tallness, capital T, is dominant over the allele for shortness, which is recessive, 3/4 of the F2 plants should be tall. And the reason why we always say should be or could be or probably will be is because probabilities are never a certainty, okay? For Mendel to get his 3-to-1 ratio or 75% tall, 25% short, he probably had to do thousands of crosses to make that happen. If you do one cross or 10 crosses, it's possible that you might not get the expected results. But because 3/4 of the F2 plants were tall, that means, so basically 3/4, it's 3/4 tall, 1/4 short. This is the easiest way to get from a fraction to a ratio, okay? So 3/4 tall to 1/4 short. You take the 3, all right? That represents this number. And then you take the 1/4 for short, that represents this number. It's a 3-to-1 ratio, tall to short. And again, these two dots, which is referred to as a colon, are represented by the word 2. The 3 represents these guys. We're talking phenotype here, phenotype, because you're going to have one of these and two of these. All of them are going to be tall. All three of them are going to be tall. 3. And then we only have 1 out of 4 that's going to be short to 1. Okay, did Mendel's results show that segregation of the alleles occurred? It did. And the proof of that is the fact that you got individuals in the F2 generation that were short. right, here we go. Whoops, sorry. Can probabilities predict averages? Absolutely. They predict the average outcome of a large number of events. And the reason why a large number of events is important, as you remember from, well, actually, I'm not sure by this point if you've seen the Bill Nye video or not, or my video of the probability lab, you can flip a coin 10 times, all right, and expect to get 5 heads and 5 tails. Maybe you end up with 9 and 1. Maybe you end up with 8 and 2, 7 and 3. 6 and 4. Okay, there is a high probability that you will not get 5 and 5 when you flip it 10 times. But if you flip it 100 times, you're probably going to get close to something like 48 and 52, or 47 and 53, or 49 and 1. You might actually get 50 and 50. Now let's take it another step. If you flip a coin 1,000 times, you are much more likely to end up with 500 and 500. And in the probability lab that I recorded a video for you, you'll notice most of the time the probability will increase from 10 coin tosses to 100 coin tosses to, I don't remember how many I'm going to do in the video. I'm going to use some past class data. I think the last one we'll do like is a couple thousand coin tosses. You'll notice that the probability increases as you increase the sample size. All right, moving ahead. A little bit of matching for you. So take a look back at number take a look back at number 20 and 21 for this. It's a homozygous. 2 identical alleles, C, or 2 of the same alleles. Heterozygous, A, have 2 different alleles. Genotype, D, genetic makeup, or the symbols that we use. Phenotype, B, the physical characteristics. Okay, because the alleles from the parents recombined to produce new combinations, meaning the short flower, this showed that the alleles, what's the best word we use to describe what happened? Segregated or separated? They mean the same thing. What's the best word? Yes, segregated. Good. What were the percentages of the different genotypes in the F2 generation of his 1 trait cross? Genotypes. If you can't remember, take a look back at number 23. And of course, I'm asking you for a fraction. So it was 25% big T, 50% big T, little t, and 25% little t. All right, so we're talking genotypes. The percentage in a probability for phenotypes would have been 75%, or 3/4 tall, 25%, or 1/4 short. Okay, probability can be used to predict. Take a look back, I believe at number 26. Yeah, average outcome of many events. Compared to 4 flips of a coin, 400 flips of the coin is more likely to produce 50-50, less likely to produce 50-50, guaranteed to produce 50-50, equally likely to produce 50-50. Yeah, of course, it's more likely to produce 50-50. It's never a guarantee. I mean, it's never a guarantee It's chance. You never know if you're going to end up with 50 heads, 50 tails. In this case, 200 heads, 200 tails. But you're more likely to get that if you increase the number of coin tosses. Organisms that have two different alleles for a particular trait. Two different alleles. Take a look back at number 20. Different, Latin root, hetero. They're heterozygous. They are hybrids. Two F1 plants that are homozygous for shortness. That means little t times little t. What percentage of the offspring will be tall? If you have two parents that are little t, you have no chance of getting a big T. So that would be a 0% chance. Okay, the Punnett square allows you to predict only the phenotypes of the offspring, only the genotypes of the offspring, both the genotypes and the phenotypes of the offspring, or neither one. Okay, so the symbols, the letters are the genotypes. But remember, the letters just represent the phenotypes that you did all the way back in step 1. So you are getting both the phenotypes and the genotypes from the cross. Ladies and gentlemen, that is it for the notes. Thanks for watching, and we will see you back in the Zoom. Why can't I stop the video?